So the process, this is MBBS question, so you have to take it home. What is the process by which we convert spermatogonium or spermatogonia to primary spermatocytes is called spermatocytes. Spermatocytes are the process or is a process or series of process that involve mitotic cell division by which we convert spermatogonium to primary spermatocytes. So what happens in spermatogenesis? The first step is spermatogonium give rise to primary spermatocytes. But before they give rise to this primary spermatocyte, let me explain something. Spermatogonium will come, divide. We say they are stem cell, and when we talk about stem cell, they are unspecialized. They are not specialized to develop themselves back or to form a specialized cell. So what does now happen? This spermatogonium you are seeing here will come and divide and form spermatogonia type A and spermatogonia type B. So what is happening? Spermatogonia type A, why is the same thing as spermatogonia? So spermatogonium or spermatogonia is the same thing as spermatogonia type A, which you can also keep on undergoing mitotic cell division to produce spermatogonium. So this type B is now what we now go and form primary spermatocyte. This process is our first mitotic cell division. Let me recap for exam purpose. Listen, spermatogonium will differentiate. They are stem cells, so they can form themselves or they can form what? A specialized cell. Now, this spermatogonium, they will differentiate to form themselves. What are the themselves there? A. They form spermatogonia type A. And they also divide, and when they divide mitotically they produce two daughter cells. So the other one they have produced is now the specialized cell we're talking about, which is spermatogonia type B. This spermatogonia type B will now perform a specific function. So what is this specific function? They will now produce the spermatozoa. Spermatozoa is sperm oh. Spermatozoa is sperm, don't forget. So let's go about to our main future. So from the primary spermatocytes, we now undergo our first meiotic first division. Now I want you to take note of something. This process begins during the puberty age or during the puberty area, and puberty time is around 12 to 13 years. Some textbook will tell you 11 to 13, but based on my research it is 12 to 13. While women or females oogenesis begins before birth. So before they give birth to females, they have already start producing oocytes, they have already start producing egg. While male, they will not start, they will only start during puberty around 12 to 13 years. Now during the adolescence or during the late puberty, which is around 15 to 16, you are about, this is the time you're expected to see your sperm, your first sperm. Your first sperm will come out before it starts becoming fluid. And when your first sperm come out, it is not expected to be that nutritious. So during your 16, 15, some maybe 14 is rare. 17, 18, 19, that is when you're expected to see your first sperm or your spermatozoa. And remember, when you are ejaculating, when you are busy doing something with a gear, you are ejaculating 150 to 270 to 275 millions of sperm. Some male can even ejaculate 500 millions of sperm. Hey, be careful, be careful. So this is your useful time and you have to make use of it properly. So let's go. Primary spermatocytes will undergo meiotic cell division. This is our first meiotic cell division. And remember, in meiotic cell division, what are we doing? We are converting 46 chromosome to 23 chromosome, and this process is meiotic and or meiosis. So primary spermatocyte is converted to secondary spermatocytes. Then for secondary spermatocyte, it will undergo another meiotic cell division. We call them the second meiotic cell division, and the second meiotic cell division is equals to mitotic cell division. Look at what I mean. From primary spermatocyte to secondary spermatocyte, what is happening is meiotic cell division, and we go from 46 chromosome to 23 chromosome. Now from secondary spermatocyte to spermatid or spermatids, we now have also what we call 23 chromosome. Why? Remember in my explanation earlier, I said that your first meiotic cell division will reduce the chromosome to 23, while the second meiotic cell division is to multiply. They are not going to reduce, they will maintain that the same 23 chromosome. Now we have our spermatid. So is another exam key question. They will ask you during the process by which you convert primary spermatocytes to spermatid is called what? So the process by which we convert primary spermatocytes to spermatid is called what? So drop that in the comment section below. So next we have spermatid. In the spermatid, make sure I want to see your response. Drop it in the comment section below. What is the process by which you convert primary spermatocyte to spermatid? Is a question for you. Just this is commonly asked question in MBBS. Drop it in the comment section below. I want to see the answer. Ngwa, let's go. I have from spermatid we convert spermatids to spermatozoa. So look at, we are converting our spermatid to spermatozoa. So this is our spermatid, this is our spermatozoa. Our spermatozoa is also called the sperm. So that process is called spermiogenesis. Spermiogenesis is the process where you convert spermatid to spermatozoa. And during this process, the spermatids are like cells, normal cell, normal cell usually have cell. They now have a nucleus, they now have Golgi bodies, they now have centroids. And what is happening? The process by which spermiogenesis is occurring. Spermiogenesis, don't forget, spermiogenesis is the conversion of spermatids to spermatozoa. So during this process, what is happening? The Golgi body or the Golgi complex or the Golgi apparatus, they are all the same thing. What is happening? It will start forming the acrosome. The acrosome will go and dissolve the zona pellucida in the oocyte. Don't worry, in the oocyte. During fertilization, I'll explain this better. Just remember that the Golgi complex form the acrosome, the nucleus form the head of the sperm. The sperm starts shedding off during spermatogenesis. The fluid in the cytoplasm starts shedding off in order to have this spermatozoa shape. While in oogenesis, the fluid is retained. I'm constructing so you have a better knowledge. I'm constructing so that exam will not trick your brain. Let's go. So remember, we are converting. So let's recap this process. From spermatogonium to primary spermatocyte, which is our first mitotic cell division. From primary spermatocyte to secondary spermatocyte, which is our first meiosis. Second meiosis is to convert secondary spermatocyte to spermatid. And I want you to go and drop the answer to this question. The process by which you convert primary spermatocyte to spermatid is called what? I want to see it in the comment section below. From there, we now convert spermatid to what? Spermatozoa. This process is called spermiogenesis.